Hello, Tuskegee University students. This is Dr. Gerald Griffin, and today I will be discussing some major themes related to our third week in Biology 315, General Neurobiology. In today's video, I will highlight major concepts in Chapter 5 of your Pervs et al. Textbook. Chapter 5 focuses on the concept of synaptic transmission. Transmission, made of the two root words trans and mission, come from the Latin mitto, mittere, giving rise to mission, which means to send. Trans means across in Latin. And therefore, the major goal of any type of transmission is to send something across. For example, this bounce pass in this game of basketball. The transmission is sending the basketball from player A to player B. This is exactly what's happening in your nervous system during synaptic transmission. It is sending something across from one cell to the next cell, or from one part of the cell to another part of the cell. Now, when we think about synaptic transmission in your nervous system, we want to point and ponder on three major ideas. First of all, what is being sent? What is the basketball, if you will? What's the message? Secondly, how is this message being sent? What is the speed? Remember, in the nervous system, we're talking milliseconds, and so we will compare fast to very fast. Also, is the message wrapped in vesicles, or is the message itself allowed to just shuttle and go back and forth from cell to cell? Thirdly, where is the message being sent? Is it to another neuron in the postsynaptic cell? Is that postsynaptic cell a muscle cell? Is it a long distant dendrite that an axon forms an autosynapse, if you will, on itself? The two major types of synaptic transmission occur from two major synapses in the nervous system: chemical and electrical synapses. Let's first discuss electrical synapses, shown here on the left panel. In an electrical synapse, two cells are actually physically connected with one another. Yes, they still have their distinct plasma membranes and they are two distinct cells, yet they are physically connected by gap junctions. These gap junction channels allow for ions and very small molecules to flow back and forth from cell to cell. And this is just moving, these ions are just moving through diffusion. In a chemical synapse, there is a space between the two plasma membranes of the two distinct cells. This space is known as the synaptic cleft. In the presynaptic cell, neurotransmitters that are traveling in synaptic vesicles are released, travel and traverse through the synaptic cleft, and then bind on to postsynaptic receptors. And remember, these receptors can be ionotropic or metabotropic. Let's take a further look on the chemical synapse, because there are a lot of interactions and activities going on here. We first want to realize here in this gray box that, again, these synaptic vesicles are highly dense, highly populating the presynaptic terminal of the presynaptic neuron. These vesicles are enriched with particular proteins called V-snares. These are the vesicle snare proteins that are going to allow for the fusion of the vesicle membrane, the vesicular membrane, with the plasma membrane. This happens through many different types of T snares that are on the plasma membrane. Some of these snare proteins on the plasma membrane include SNAP25 and syntaxin, as well as synaptobrevin, shown here in pink. When SNAP25 and syntaxin bound together, they allow for a major complex to form. When this complex forms, synaptobrevin is pulled into play and allows for the interaction with the vesicular membrane. When the synaptobrevin and the complex forms here from the plasma membrane, it pulls that synaptic vesicle, this circle here, closer and closer to the membrane. We now say that the membrane has docked. This is the docking phase of chemical synaptic neurotransmitter release. When we have an influx of calcium through voltage-gated channels at the presynaptic terminal, that means that these calcium channels are only going to open at the appropriate depolarization. The calcium allows for the direct physical interaction between synaptotegmin and this major complex. When this occurs, the vesicle gets so close and is able to fuse with the terminal plasma membrane. And when this happens, this allows for the release, the exocytosis of neurotransmitter. Now, every vesicle in a presynaptic terminal is going to have the same amount of neurotransmitter. So thousands, tens of thousands of neurotransmitter molecules are going to be in discrete units. These discrete units are contained in the vesicles. This is what we know as quantal release. That every vesicle contains some quantum, some discrete quantity of neurotransmitter molecules that can be and will be released upon appropriate stimulation. Let's go to a quick animation video depicting neurotransmitter release. Going to enlarge the screen here for you. And now we're going to also focus on synaptic vesicle cycling. If we play the video here, this short animation, you see the first phase is mobilization, docking, then fusion, and then release, and then recycling. Remember, first the vesicle will mobilize. It will move closer and closer to the presynaptic terminal. When the snare proteins, the V snares and the T snares bind, they're going to pull closer and closer and will dock. When calcium comes in, it allows for the fusion and the release of the neurotransmitter. Now that membrane, and I'm going to keep playing it, that membrane of the vesicle is going to fuse back up and it'll get recycled back and then get appropriately filled with the correct neurotransmitter and is allowed for more release upon further stimulation. I put the website here, which is just neuroscience.uth.tmc.edu/s as in Sam 1/chapter05.html if you'd like to view this website for yourself. Now, back to this presentation. Hopefully, you have had some major ideas and are able to solidify and make sense of the mechanisms allowing for both electrical synapses and chemical synapses. Well, why do we care about synaptic transmission? Well, it is the way that the message, whatever that is, gets carried out. So whether you are perceiving your environment, first sensing your environment, or perceiving your environment, or reacting to your environment, all that happens at the synaptic level. In addition, there are some major disorders that arise when we actually have synaptic dysfunction. One of those is myasthenia gravis. In myasthenia gravis, it is mainly thought of as a muscle disorder. Individuals have hard time and difficulty in breathing, walking, maintaining balance, and it can have severe effects on the quality of life. In fact, if not treated, it can be fatal, mainly because of the issues related to breathing due to the fact that the muscles in your chest cannot contract properly. Moreover, there is a specific type of myasthenia gravis that affects the eyes, and this can actually lead on further to full myasthenia gravis. Myasthenia gravis is an autoimmune disorder. And so these muscle disorders, muscle symptoms, if you will, are all due to the fact that in individuals with myasthenia gravis, they actually produce autoantibodies that actually will bind to the acetylcholine receptor. Acetylcholine is particularly important at the neuromuscular junction, where the nerve is going to synapse onto muscle cells, causing for contraction. Well, if we have autoantibodies to the acetylcholine receptor, even if we release acetylcholine, the neurotransmitter itself, the neurotransmitter cannot bind to the receptor properly. In fact, in one instance where we actually cause motor cell destruction, you actually see a whole wiping out of the muscle membrane. Another way that myasthenia gravis could occur is that this autoantibody could cause cross-linking of the acetylcholine receptor, causes the receptor to internalize and become degraded, and therefore not allowing for any further muscle activation. And lastly, the autoantibody could bind directly to the binding site, acetylcholine's binding site on the acetylcholine receptor, and acting as a competitive antagonist, not allowing for any acetylcholine to bind and leading to a major disruption in the ability of this muscle to contract. This is the importance of actually understanding how synapses work, their structure, and how function and dysfunction and function are related. So, the goals for week 3, and this is related to chapter 5 in neuroscience text by Pervs, is that you are able to differentiate advantages and disadvantages of the two major types of synaptic transmission. You need to also be able to apply foundational mechanisms of synaptic vesicle release and cycling to pharmacological interventions for major neurological disorders. This is the only video for week 3 that you need to watch. Please contact me if you have any questions, and I look forward to working with you for the rest of this course. Thank you, and have a great day.